Hi, I'm Dr. Adrienne Gorny, and I'm a plant nematologist in the Department of Entomology and Plant Pathology at North Carolina State University. I study root knot nematodes in sweet potato and other crops. Specifically, I study these nematodes and look for new and better ways to manage these important pests. Today, I'd like to share with you how scientists and nematologists identify Meloidogyne enterolobii, which we abbreviate as ME. You see, root knot nematodes belong to the broad genus or broad group of nematodes called Meloidogyne. But within this broad group, there are species or very specific types of nematodes. ME is only one of these species. But it's very important that we be able to tell the difference between ME and closely related species. Scientists have two methods for identifying ME: morphological and molecular. Morphological identification is when we look at the nematode under the microscope and determine what species it is based on the appearance of the nematode. Here you can see the head, tail, and worm-like body of the nematode. But the tricky part is that many root knot nematode species look highly similar under the microscope, and so therefore it's very difficult to tell them apart. This is where molecular identification comes in handy. Molecular identification is when we analyze the DNA of the nematode to determine what species it is. Just like all living organisms, ME has unique DNA that makes it different than other living organisms. In the lab, we extract this DNA and analyze it to determine if it matches that of ME. This molecular method is highly precise and accurate compared to the morphological method, but it does require careful scientific training and specialized equipment. First, we extract DNA using a special detergent. Then we mix this DNA with a compound that makes copies of it, almost like a photocopier makes copies of a paper document. In order for this compound to make copies of the DNA, we have to place it into a machine called a thermocycler. You can think of a thermocycler as a special oven, which we incubate the DNA at a very precise temperature for a very specific amount of time. Once done in the thermocycler, we take the mixture and place it into a gel-like substance and illuminate it with a black light. As you can see here, these lighted bands indicate the presence of ME. As you can see, identifying ME can be a tricky task, even for experienced scientists and nematologists. But it's still very important to distinguish ME from other root knot nematode species. If you think you have ME in your field or greenhouse, or would like help identifying ME, or would even like more information about our FINDMe project, you can send us an email at findmenematode@gmail.com. You can also learn more about our research project at findmenematode.org or interact with us on Twitter using the handle findmenematode. Thank you for being part of the solution.